see, we are going to learn about molecular and cellular biology. This module is introductory. So the last lecture was just introductory, and in today's lecture we will learn more things about the cell. And if you think about cell, it can be considered as the basic unit of the life, because you consider any organism, it would be consisting of cells, and depending upon whether organism is unicellular or multicellular, the number of cells will be different. So when you think about the living being, you can ask me one question that what would be the thing about the virus, whether we can consider virus as a living being, probably not, and it can be only come to life when it is basically interacting with any live object. Until that time it is dead. So it's a not completely living system, and it has a little different mechanism for reproduction, or it uses the host system to getting more numbers. But when you consider about the cell and its discovery, the cells were discovered with simple microscope, and even when you think about the unicellular versus multicellular, the unicellular organism can display certain multicellular-like behavior under some conditions. So in today's class, here are the learning objectives. First, we will learn the surface area to volume ratio maximization, why it has to be maximized, and then the compartmentalization of the cell, and level of the organism, metabolic rate. So all those things are somehow related to the surface area to volume ratio. Then we will talk about the reductionism of cell, and how we will basically study cell, because it's a very complex. We can break it down, and then after breaking it down, one can reduce the complexity. Then similar like manufacturing unit, there are basically inside cell there are manufacturing units. Those are basically ribosomes. We'll talk about that. And then there are something like scavenging bodies inside the cell. Those help to digest the food and damage organelles. Then, in order to support the cell structure, we have very specialized organelle called cytoskeleton, and because of that, cell can take the structure. And apart from that, we have another important protein family that is called as you can say the proteins which can move the cargo or move the protein or any other biomolecule from one point of the cell to the other point of the cell, and those are basically motor proteins, and that is basically something referred to as intracellular cargo. So these are the objectives we are going to talk about, or we are going to discuss about. Now, I will basically have a lot of emphasis in this lecture about the surface area to volume ratio, why it has to be minimized for particular on the cell. So here we'll give some analogy. What does the surface area to volume ratio, and how exactly that could help us to understand things happening inside the cell. So here what you see is there are three tubes. First tube with unit length, the other the length has 5, and the third one basically we have subdivided this big tube with the length 5, but then it is subdivided into unit tube, and in the third one there are 125 small tubes. But here In the third one, what we are going to do is we are going to ignore the small spaces, small space in between the cubes. And when you consider about these three circumstances, these three cubes, and you calculate the surface area, for the first one with unit length, the surface area would be 6. For the second one, which has the length of 5 units, it will have surface area of 150. And if you think about the third one, there are 125 cubes with the unit length, and hence the total surface area for this particular instance will be 750. And corresponding volume can be seen. So for the unit length, the volume would be 1 cubic. For the second one, it could be 125. And then the second and third, you can see the volume for second and third would be very much similar. Then when we compare the surface area to the volume ratio, for the first one that comes 6, the second one that is 1.6, 1.2, and the third one is 6. So the question is basically, could the surface area to volume ratio has to be maximum or minimum? And that basically depends on the situation. There are some, day-to-day situation where you can see the surface area is more and the volume can be basically the surface area to volume that can be reduced. So in the first case, what you say is this person is sipping tea in the saucer, and this, the adjacent person who is that person is sipping tea with the surface. In this case, when the person is sipping the tea with the saucer, what's going to happen is because the tea is exposed to larger surface area, it will cool down faster. So probably this person likes tea with little less hot, and this person may like the tea with lot of hot tea. So depending upon the choice, you can see the surface area will come in picture, and that would help us. The second picture over here is for the room heater, and you can see there are pins, and these pins are kind of stacked. And with this kind of design, what you actually gain is you are basically having a lot of surface area exposed. And with the lot of surface area exposed, what happens is the heat is being exchanged faster in order to have room warmer faster. The third one, you can see large ice cubes. And the fourth, this is ice cubes which are one can use in the soft drinks also. But then let's say given a situation, let's say you want to cool down fish, or you want to cool down soft drink, or any other liquid which is potable. Then what size you will have? Will you use small cubes or very large blocks of the ice? So in consider when you have seen the fishermen, they have the special ice boxes, so big ice boxes, and in order to keep the fish cool, what they do is they use larger ice box, larger ice stacks also. And simply, this is again, the concept of surface area to volume ratio. And for this one, the soft drink, what you want to do is you want to make the ice melt faster, so that the volume, typical volume, should get cooler faster. So these are different sizes, and one can relate the concept of minimizing surface area to volume ratio. So when you think about the cell, there are different cells. You can say bacterial or eukaryotic cell. So this is mycoplasma, one of the bacteria. It's very small. The diameter of this bacteria is of the order of 0.1 to 1 micrometer. Then typical bacterial cell is a little larger, and it could be 1 to 5 micrometer. And in case of eukaryotic cell, it is little larger than the bacterial cell, and that could be 10 to 100 micrometer. So when you think about this diameter across the bacterial or eukaryotic cell, you can see the magnitude is almost like 100. The magnitude change is 100 times or so. So if you consider that, then let's just consider a human being, a bird, a fish, and in this particular case, you think about the cell, and the cell of each organism in this case is much smaller than the relative size of the organism. Do you wonder why it is so? And there is a particular reason to it. This basically, the idea is again, saying you want to minimize the surface area, the ratio of the surface area to the volume. And let's just talk about in some scientific way. So you can consider cell as a thermodynamic open system, and when you think about that, since it's an open system, it will exchange heat and matter with its surroundings. And in order to have this exchange, it will do some work, and if cell is doing some work, it will require some energy. Apart from that, cell can uptake some nutrients and metabolize them and excrete waste products. So basically, you can see the work. In order to do this work, it requires the energy, and the heat is basically a form of energy. And when you think about nutrients and the waste products, that is basically matter. And both of these things are being exchanged with its surroundings, and that is why we can consider cell as a thermodynamic open system. Now let's just come back to this concept, maximizing surface area to the volume ratio. So this particular situation in the cell helps in efficient exchange of heat and matter. And larger organism does not mean larger cell. It just it has a more cell, and size of the cell remains pretty much similar, or you can say it would be pretty much same. There could be some few exceptions, but the idea is very clear that in order to have sufficiently high ratio of surface area to volume, then in that case what happens is cell can have exchange of a lot of material with its surroundings. Specifically, you think about the intestinal cells. Over there, you want to digest food, and approximately in a one meal, one person can have, let's say, about 250 gram of content, whether it's dal, chawal, roti, whatever is veg, non-veg. I'm just giving some number, and then you imagine if you have that much volume, you have that much 250 grams of food, and you want to digest it faster. Ideally, that food should expose to intestinal wall, and if you have seen, you might have seen that intestinal wall, that is kind of, it has a very peculiar design. It goes zigzag manner, and so the zigzag manner is to basically increase the area, and so that since there would be increase of the area, and it can get in touch with more food, and that process, the digestion process, can be a little faster. And apart from that, there is some cells might have very specialized function, and with that specialized function, again having this ratio maximized, that helps. So then but then whether there could be any upper limit for this size of cell. Theoretically, we can make cell let's say 2 meters long, but will it help us? Probably not. So you have seen that most of the cell will have of the order of micrometer also. So that is there is a reason, and you might not see the cells which are larger than the micrometer, except neuron cells. And this neuron cell, again, they have very different structure. But again, with when you think about the neurons, some neurons are 1 meter long. But even though they are 1 meter long, still, the this area, surface area to the volume ratio that has been maximized. We'll come to that after some slides. But that's one of the exceptions. But when you think about cell size, probably the metabolism, the rate of metabolism, that will set the upper limit of the size of the cell. As the volume increases, you can notice that when the surface area is also basically when volume increases faster than surface area, then organisms prefer having many smaller cells than having only one single larger cell. So that again the manner in which volume is going to get increased, in the similar manner the surface area of that particular cell is not going to be increased. So again, when we think about this situation, when we think about the metabolism, this is very much important. Maximizing surface area to the maximizing this particular resource surface area versus the surface area to the volume. So here we are just again thinking from the thermodynamics point of view, and let's just say we have inside there is a many reactions are going inside the cell. Just here we are taking very simple reaction. This is the reactant, and this reactant is being converted into product. And if you think about the free energy change, that will be given by this formula: delta G is equal to minus RT log K equivalent plus RT log of the rest of the concentration of product divided by concentration of the reactant. We are not going to discuss this much, but then the idea over here is again to have this faster metabolic rate, you need to have this ratio maximum, and so that is basically main idea behind having this cell. So again, coming back to the typical sizes of the cell, and thinking about the prokaryotic cell versus eukaryotic cell. So you can see the prokaryotic cells are much smaller than the eukaryotic cells. So then the obvious question would be the volume for the eukaryotic cell is larger, but then how does this particular eukaryotic cell will perform, and what kind of strategy it will have to increase the surface area and increase this particular ratio? Then the answer over there is inside the cell you have a lot of cellular organelles, and they have very specialized functions. And when they do this, I mean, again, you think from the metabolic point of view, within the cell, there are many activities going on, and in order to have that activity faster, the organelle will have this ratio maximum, and we will come to that in a little bit. But when you think about prokaryotic versus eukaryotic, the simple analogy is you can say 3-bedroom flat versus 1-bedroom flat. So if you think about the 3-bedroom flat, you have very specialized compartments. You have a living room, you have dining area, you have the porch. Then there could be different bedrooms, and depending upon situation, you may have a garage or so, and then you may have a kitchen. But so this could be your eukaryotic cell, and in the one-room flat, practically you have all the functionalities available. you will have kitchen, you will have bedding area, you will have kitchen area, dining area, but then it's just a small room and everybody is living happily. Again, with the nature, this kind of things are there. So with this two situations, there are some advantages and disadvantages in each case. So when you think about one room flat or one room kitchen, everything combining together, some of the advantages are it's easy and cheap to maintain because it's a smaller size and it can adapt easily to the situation. And just in case it has to move to a different location, this person can easily pack and they move very easily. But then at the same time, the biggest disadvantage would be there is a lack of privacy, and obviously there is a small space. Within the small space, everybody is confined. So compared to that, the three-bedroom flat. Over here, what are the advantages? The biggest advantage is basically you have a compartment for each activity, and there is a privacy and independence for the activities. But at the same time, when you think about the disadvantage, biggest disadvantage would be expensive to maintain, and it is difficult to adapt, and it will have a lot of constraints while moving from one place to other place. So, so when you think about this prokaryotic cell, you see it's a single cell, and within single cell, you have everything over there. So, if you think, now we'll get into a little bit terminology, and these terminologies are very much new, right? So, we will try to understand what is the function of each part of the cell. Prokaryotic cell. So if you look through it, the outer you have some kind of hairs coming out, and those are called fimbriae. And these fimbriae are very much important in terms of attaching. So this structure is important to adsorb on some surfaces, or it can adhere to the adjacent bacteria or adjacent animal. So it kind of provides the anchor for this cell. Then you have the nucleoid over here, which is shown in the purple or blue color also. And this nucleoid is the region where you can find the DNA. But then this is not enclosed as such. It is, it is not enclosed by the membrane. And then you have, see these dots are over here. You can see a lot of dots over here. So those dots would be nothing but you can say ribosomes or some other particles. But let's just say that those are ribosomes. And the ribosomes are basically the protein factory for the cell. What do you mean by protein factory? So protein factory is basically ribosomes are the molecules inside the cell which can prepare the protein. And when you think about the evolution, probably the protein didn't come first. It was the ribosome that acted as an enzyme. And then, from the ribosome itself, everything, the earlier cellular structure didn't have any protein or access for enzymatic activity. And it was, it used to call as ribozyme. So, that's the historic point of view. But apart from that, we can see over here there is a cell wall, and the cell wall is rigid structure outside the plasma membrane, and then you have, okay, so inside is the plasma membrane, and the plasma membrane is encloses basically this plasma membrane encloses the cytoplasm. The cytoplasm is inside everything of the cell, and then just to just adjacent to this plasma membrane or outside of this plasma membrane, you will see cell wall, and it is the rigid structure which protects the cell, and then there could be a capsule to it, and this capsule is nothing but something like, I can say, jelly-like structure, and it will have extra layer of a coating also. And then you see these are the long hairs over here. Those are the flagella, and these flagellas basically that can propel the bacteria, and the bacteria can move from one point to the other point in search of food or in search of anything basically. And on the right side, what you see is the image obtained by transmission microscopy of this particular bacteria that is called as Bacillus turbulens. Now, when you think about the eukaryotic cells are very, very different. And what you notice is there is a lot of organelles over here. And then you might have a very similar thing, like ribosome is there. Then you might be able to see this blue structure, green structure, and then some other yellow structure over here. So they are very, very different than the prokaryotic cell. But then what you see over here, all these structures are kind of in zigzag pattern, or they are, they have some bending, they have some curvature. So this curvature is again what it shows that it basically increases the surface, it maximizes the surface area to volume ratio. Even though, and that's when, now you can basically relate that, even though the cell size of eukaryotic is much larger than the prokaryotic, at the same time, it has to have metabolic rate faster. It has to have a lot of, metabolism going on inside the cell, and it has to maintain its status quo. So in that case, the strategy adapted by eukaryotic cell inside this, the organelles, the particular organelles, again, that would have a lot of curvature, and with the lot of curvature, it can have exposed to a lot of surface area, and since, within the same volume, you can have maximization for this particular ratio. So we will think. I will come back to this apparatus, and I will talk about all this nucleus and all those things in a while. So over here, what you see is again the blue part over here, that is endoplasmic reticulum. Over here, the Golgi apparatus. You have the mitochondria in this, and then you have the microvilli. So all the structures you can see kind of either tubular, or it has a cylindrical shape, or it has a curvature. Again, so basically you can think about speed over here. If you have larger pore, only larger particles can be separated. But then so the idea over here is to again maximize your ratio of surface area to the volume. So now considering this organelles, so this is the nucleus, and inside nucleus there will be DNA, and then apart from that DNA, there will be some other things happening inside, and over here you can see some pore-like structures are there. So basically through this pore, the nucleus basically, inside nucleus there is a DNA. DNA will get eventually transcript into the RNA, and then RNA will come out of this the pore-like structure. Those are the, you can say, membrane proteins also. Again, the nucleus is kind of inside the special membrane, and it is protected from the cytoplasmic thing, and just next to the this nucleus, you will see this blue structure, which is endoplasmic reticulum, and endoplasmic reticulum has two sides, inward and outward, or you can say pivot fan. So there are basically uh, uh, there are rough side and uh, smooth side of the endoplasmic reticulum, and the rough side, you can see over there, the ribosomes are sitting over there, and it basically the main function of the endoplasmic reticulum is to biosynthesis, and we'll talk about this all these organelles in a while. So moving to the next concept, and when you want to study the cell, you want to basically have different techniques. So there are when you think about finding out what exactly cells look like, you have to go for the microscopy technique. And there are different microscopy techniques. In the light microscopy, you will have super resolution microscopy, confocal microscopy, and there are many things over there involved. Apart from that, recently some advances are happening in the electron microscopy, and with that, those electron microscopy, one can get the structures of atoms at the atomic resolution structures of until 3 angstrom or 4 angstrom or so. So when you think about light microscope, one can have the resolution up to 10 nanometer also. And most of the cells are basically, it has the diameter of 1 to 100 micrometers. So one can easily see the cell with the light microscopy, but in order to understand what is happening inside, you want to have a little better resolution. And that better resolution can be provided by different techniques. So that's where we need to have the super resolution microscopy, which can further improve the resolution. Now, when you consider about the microscope, there are three parameters which are important. One is the magnification, other is the resolution, and the third one is the contrast. So magnification is just you can say the ratio between object image size to its real size. And when you think about resolution, it is you can say measure of clarity of the image, and it is the minimum basically resolution. It's kind of a description, or by the resolution criteria, one can estimate what is the minimum distance between 2 points can be separated, and still you can distinguish those two separate points. The third parameter is the contrast, and the contrast is basically, you can think about the difference in the brightness between light and dark areas of the images. Here what you see is one cell, so it's a chicken egg. At the starting, you can think that chicken egg, it has only one single cell, and from this single cell, or you can say it's the egg inside this. When you think about the single cell, and what happens is this inside this there is a lot of albumin which can serve as a food for the single cell inside this egg. And when it is fertilized, what happens is it forms the embryo, and from embryo it basically divides into the multi cells also. But then starting from the one cell to multi cell, it has to think, nature has to think about how to again minimize the surface or maximize the this particular ratio, surface area to the volume. And again, having that maximization will help in the metabolic activities happening inside. And during the hatching process, you can see that the cell will require a lot of food, and it would again it would interact with its surrounding environment, and with that it will have lot of exchanges of the gases also, or richness of the matter. The second example over here is the nerve cell, and the nerve cell basically is a very unique situation. The smallest nerve cell could be up to 1 micron or so, but the biggest could be up to meter or so. But at the same time, you can see the cell has a lot of cylindrical structure or tubular structure. And this particular tubular structure, even though the length is higher, and with this kind of structure, it is maximizing its surface area to volume ratio. So again, when you think about the organisms, animals, they have different body mass, and every organism might need different chemical energy, a different amount of chemical energy. The larger animals will have more body mass, and they will require more chemical energy. This part, that would basically, then you have to think about the metabolic rate, whether the metabolic rate will get affected, or the ratio of the body mass and then chemical energy will that affect the metabolic rate. So the energy required to maintain each gram of body mass is inversely related to the body size, and you can think that each gram of mouse requires somewhere about 20 times as many calories as the gram of the elephant. So just it's some kind of comparison you want to know, but the idea over here is the same. There is always some kind of trade-off, and in order to even though the body mass is higher, it will have more metabolic activities, it will have more number of cells, but then in order to carry out this metabolic activities and keeping this organism alive, always there would be strategy that it would try to maximize this ratio. Excuse me. Now here, I'm giving you one more example, and I think I already talked about the intestine, so it's a very similar example. So what you see is it's a python which is following the earth, and once you might have heard or read somewhere that once python has its prey, after eating it is basically very lethargic. It will stay at that particular position for a long time. And during that process, it is digesting. So again, for the digestion, you can see the inside structure will be very much different. And the inside of this python, again, it will provide such a ratio, the surface area to the volume ratio is maximized. And due to that, it can digest the deer. But again, the digestion may happen over a week or so. So now the next concept or next objective we want to learn or discuss is about cell strategy, or how to study the cell. It basically approach what we do is reductionism. So again, cell is very, very complex system, and in order to understand what we do is we want to break it down, and once you break it down, we will reduce the complexity. So, I'm just giving you one analogy over here. So you have seen this, you might have seen this device, and over here, this is a mechanical device which can hold few things. Particularly, this will be handy in terms of when you want to change the flat tire also. It provides support, and at the time you can see this device in workshop, where it's workshop. So when you want to, when you buy the device, it comes along with its instructions. So those instructions will tell you which part goes where and how to assemble whole thing. So again, if something goes wrong, then you have some, this, you go back to this manual and see, okay, this part is missing, that part is missing, and you try to deal with that. But other than that, this is, you can say, simplest assembly which human-made has done. But apart from that, if you think about the shuttle rocket, it's very complicated. And when astronauts are traveling in the space, then you can see all the basic amenities has to be provided within that rocket. So that could be very complex system, and you have to basically, again, over here, this is man-made. You have created it. So it would come along with the manual. So you know how to assemble this part. And again, it could be complex, and this shuttle rocket would be very, very complex. But when you think about the nature, so all these different organisms are there. And this will have different parts, components, and you have to know how many parts are there. You have to know what is the sequence of assembly. It's extremely difficult to predict this thing. So when you think about this living being or organism, they are basically, you can say, self-assembled system. And this self-assembly has, it has basically evolved since millions years. And in this basically self-assembly process, some parts will come, some parts will go. At particular stage, that organism might need few things. Again, if you think about the appendix part in our human body, appendix as such doesn't have any function, but still it is there, and we really don't know why it is there. So, but then during, let's say after 10,000, 20,000 years or so, this appendix might vanish from the human body. That's a other process. So how basically cells, organisms evolve, that's a very complicated system that comes in the development biology area. And but over there, the idea is, I'm giving you analogies over here. This is simple human-made object which comes with the instructions how to assemble it. But when you think about the living being or organism, we don't have any instructions for the assembly. Very complex. So then, in order to get this component inside the cell, how do we obtain this component? And then there would be two questions. So there are, so many types of organisms, living beings. Then do you want to study all of them in order to understand the particular component of the cell? Or you want to basically study only a few of them? That's the first question that comes across the mind. The second question is basically most cells are small in size and not visible to naked eyes. Then how do we study these objects which cannot be seen by naked eye? So now, you have the choice of experiment. And again, I'm giving you one analogy in the nature. So in the first class, we have seen that there are two two organisms, and basically we have seen this cilia, and cilia structures are very much similar. Architecture is very much similar, and they have very different functions. So when you think about the model system also, and when you think about the nature will find very optimal solution, and it will retain something over million years of the evolution, and it will adapt those particular things in different manner. So even though the structure of the cilia is very much same, architecture is very much same, but then the function is very much different. So I will make a point of this, in a minute. So, in order to have, model systems, or in order to understand the cellular organisms, you can study few model systems, like E. coli. So, like in case of bacteria, E. coli is very well studied. Even E. coli is common name, but then the full name is Escherichia coli. Then most of the developmental biology labs, where they want to study the biology and how exactly the embryo to the full organ happens, and what are basically developmental stages. The favorite organism is the zebrafish, and then you have the sea lichens, weed. So these are the model animals or model systems. Over here, you study this, and then after studying this model system, one can know the components of the cell. So to answer the first question, it will suffice to study few model systems, and which are easy to work with in the laboratory, in terms of because those model systems will be easy to grow, and it will have short generation. And even though you have the model system, but remember that there are few organelles which might have very different functions in different organisms, but the structure will remain very much similar. And the second question, most of the cells are, to answer the second question, what you do is you basically go for this reducing the complexity of system by breaking the system, and by breaking the system, you can simplify it, you can understand how it works. And this way, we can have this principle called reductionism. So, in order to break it down, how do we basically break it down, and how do we basically study it? So, the simplest concept is you can take it. You can think about the physical phenomenon, and you can take the contributions from the physics, chemistry, and engineering. Over there, what you do is you can use centrifuge, and with the centrifuge, there could be various centrifuges depending upon its speed. So, simple is tabletop centrifuge. Then you might have high speed centrifuge and ultra centrifuge. And with this kind of different centrifuges, one can go up to the force of 20,000, 200 K. So this is basically gravitational force. And now apart from that, we have one more technique that is called microscopy, and over there, biologists can help us to find specific organelles. They can mark, or they can prepare specific assays, and there could be again different techniques in the microscopy. It could be light microscopy, scanning electron microscopy, fluorescence microscopy, transmission microscopy. So now here are some representative images obtained by various microscopy techniques. So this is bright field, bright field image obtained by bright field microscope. Then you have specific technique which is called as phase contrast. Over here you can see the objects are very clear, and then the contrast is very clear. So almost same, 1, 2, 3, they are basically same images. Over here, you might not be able to see anything over here. In the bright field, you are able to see some cellular structure, and in the phase contrast, you might be able to see some subcellular structures over here. So these are different techniques. Then with the fluorescence, what you do is you can have a fluorophore attached to particular protein, and it will have some color. It will imitate with that color, and that color can be captured with microscope or detector, and then then you can basically map it, and after mapping you can get this kind of images. Then you have the confocal microscope. Then apart from that you have the super resolution microscope, and then you can have the electron microscope, and within the electron microscope there are two things: scanning electron microscope and transmission microscope. So depending upon what point you want to study, how minute or how microscopic you want to study, this technique will vary. And light microscope with the super resolution, one can have the resolution of the order of 10 nanometer also. And beyond that, we want to basically go for transmission electron microscope. I'm here, going to show you how cells inside the cell, the objects inside the cell can be captured with cryotomography. So this is it's kind of reference imaging technique. So here what you see is the budding events in the SARS-CoV-2 viruses, and it is interacting with the membrane, and it is getting fused and getting engulfed. So there are a lot of events over here happening, and in order to get that kind of images, get this kind of understanding, over here, what scientists do is they record this particular in terms of volume, and this is the volume will look like, and you can see at the different it is basically focusing and defocusing at different levels, and with that one can see different subcellular organelles, and you can segment it. After segmenting, you can identify these different structures, and you can mark it. So that's how this subcellular structure can be revealed. So the other technique is the centrifugation, and for that you want to go for cell fractionation. So centrifugation basically that will separate the various components of the cell depending upon the its density or its size. And when you think about microscopy, microscopy will identify the components in each this density or its palette. So and apart from that, you can apply biochemical methods to determine the metabolic functions of each fraction or palette. So over here, what you see is this is particular cells. They are let's say over here there are tissue cells with different organelles, and what you do is you go for homogenization process, and with the homogenization process, after that you can centrifuge it. So for the centrifugation, what happens is at the lower speed you can separate out the larger components, and at higher speed you can separate out the smaller components of the cell. And the fractionation of the cells will enable enable specific cell components to study, and then so at different speed over here you can see you are able to obtain different parts of the or different organelles of the cell. So somewhere about 1000 G course one can get the palette reached in the nuclei and cellular debris. Somewhere about 20,000 G one can get the mitochondria and chloroplast. Then somewhere about 80,000 one can get the microsomes, and then higher speed one can get the ribosomes or some DNA particles, RNA particles also. So at different different centrifugation speeds, this particular technique is called as differential centrifugation. And at the 10 minutes, I mean, you first what you do is you run the centrifuge for 10 minutes at this speed, 1000 G. Then you obtain the pellet, you separate the pellet, and then you go for the next centrifugation run, and in that it will have let's say 20 minutes at 20,000 G also, and then the process will continue. So at each stage you will get different organelles, and with biochemical methods one can study these organelles. Now, coming back to the you know the cellular structure of the eukaryotic cell. So when you look through it, this is you can see lots of things are there inside the cell, and so in order to have this, all these organelles will have their own specialized function, and it has the ability to interact with other parts in coordinated manner. So particular shape and size of this these structures are there for specific reason, and it's not that it happens arbitrarily. The main concept over here is again it tries to maximize this ratio, surface volume or surface area to the volume ratio. So now when you think about the cell has many things happening inside, and in order to survive the cell, it will have some kind of manufacturing process. It will have some kind of different activities, which is very similar to manufacturing, packing, and delivery. So there could be these are basically some mobile proteins, or we can say mobile biomolecules, and then apart from that there are proteins. So proteins are very essential for the living being, and whatever work is done inside the cell, that is mainly done by different proteins. So here again I'm just going to give you some analogy. When you think about this, this is cellular, this is mobile manufacturing unit in the USA. You can see there are different lines over here, and at each line there may be some specific function. Somebody might be in this line maybe just doing the work on the camera. Somebody might over here just fixing the chip, and then somebody might over here they are basically fixing everything together and making the final packaging. And when you have each line very specific function, you can work in coordinated manner, and you can have your productivity higher, just like and so. And then after that, when you manufacture the mobile, it has to be packed, it has to be shipped. So there could be a particular process to it. How do you pack it, and from packing to shipping to reaching the customer, there is particular process. I mean, every shipping company wants to minimize the time. There will be a lot of protocols. Similar like that, inside cell, a lot of things are being made, manufactured, and just not that, after making this thing inside the cell, it has to reach to its particular destination. And now coming to the the particular parts of the cell. The first part I'm going to talk about the endoplasmic reticulum. So over here, endoplasmic reticulum is biosynthetic factory of the cell, and basically it is has a lot of process, and when you talk about the this endoplasmic reticulum, it can synthesize lipids, carbohydrates, then it can store some minerals, something like calcium, and it can help in detoxification of some drugs and some poisons. So when you want to understand this word endoplasmic reticulum, this is what endo means within. Plasmic will mean the cytoplasm, so within the cytoplasm, and reticulum is basically network. So if you look into this figure over here, you can see there is a kind of network, and that is how this the terminology comes. When you think about the terminologies in the biology, either it will have Greek or Latin origin. Most of the time, these two the words from these two languages are taken, and that's where the terminology comes up. So the endoplasmic reticulum has two basic things: smooth endoplasmic reticulum, and then you have the rough endoplasmic reticulum. And when you look inside the cell, this endoplasmic reticulum will be just nearby the nuclear envelope. So this is your nuclear envelope, and the endoplasmic reticulum is encompassing this nuclear envelope. So these are the lumens of endoplasmic reticulum, and then there are cisternae. The red spot over here, they are ribosomes, and then you have upon maturation, what happens is this it will basically have this vesicle formation, and this vesicle will get transported towards the Golgi apparatus. That we'll talk about it in a bit. So there is a lot of process happening over here. The endoplasmic reticulum, basically it can have the metabolic activities on the both sides of the lumen. There could be there could be specific things which are happening, and then it can transport the protein in outward direction also and inward direction also. So when you think about the lumen, it is enclosed space. The systemized layer pack, and again, the endoplasmic reticulum is you can see it's continuous with the nuclear membrane or nuclear envelope. So yeah, so in terms of the its activity, smooth endoplasmic reticulum will have the biasing of lipids, metabolic. It can basically metabolize the carbohydrates. And when you think about the endoplasmic, rough endoplasmic reticulum, particularly, it is very much important for the protein, and it can synthesize, and the ribosome will sit on this endoplasmic reticulum to synthesize, and then it may enter the endoplasmic lumen through the pore for the modification. That, I mean, again, there are there are once a protein forms from the ribosome, so there could be some modifications over there. The next organelle I'm going to talk is about the Golgi apparatus, and it is basically you can say it receives the molecules from the endoplasmic reticulum and ships across the cell. There are two phases. We have the cis phase and trans phase. So cis phase is the receiving side, and trans phase is the sitting side, and you can see the cisternae over like this. Unlike endoplasmic reticulum, these cisternae are stacked; they are not connected. In the endoplasmic reticulum, the cisternae are stacked, and this is typical figure or image obtained by transmission microscopy for the Golgi apparatus. So in initial days, it was thought that before going to this, I want to discuss two things over here. So there are vesicles, and or you can say these are vesicles over here, and that can move from endoplasmic reticulum to Golgi apparatus, and upon maturation of this cisternae, the Golgi cisternae can move in cis to trans direction, and this vesicle, it can basically leave the Golgi apparatus, and it can carry few products or proteins, and that can be reached to its destination. So these vesicles again can serve as a transporter for the protein. But then there could be bigger questions over there when this how do you basically know that this destination this vesicles has to be go? How do you basically move the protein from one point to other point? And for that there are some different mechanisms, and we will talk about that in a bit. So now the next objective is you know how does cell manages to uh, digest or to digest the food or whatever the scavenging activity is happening over there. How does it manage it? So for again I will come to the analogy over here. Let's say when you think about stolen cars, they are taken to the workshop where they are dismantled, and after that dismantling, whatever usable parts are there that can be sent to the spare parts to the dealer, and unusable parts are sent to the scrap. Just like that, inside the cell, there could be, since a lot of metabolism activity is happening inside the cell, metabolic activity, there would be synthesis as well as there something will get down broken down. So and during the breaking down, some parts will be usable and some parts will be unusable. Then how do you deal with that? So to deal with that, you have lysosomes inside the cell, and these are particularly called as digestive compartments. And they are kind of circular pattern, and it has particular enzyme called as hydrolytic enzyme. Okay, and when you consider the lysosome, there are two main activities for the lysosome. One is phagocytosis, other is autophagy. In the phagocytosis, what happens is the food vacuole. I mean, there are food vacuole, and that basically, that food vacuole has the food, and that is basically getting digested with the lysosome. So lysosomes will have this, you can say, digestive enzymes or nothing but hydrolytic enzymes. And after digestion, what will happen is these food particles will further disintegrate, and then with that disintegration, you might be able to get the micronutrients, proteins, amino acids, which would be very much needed for replenishing your body. And apart from that, you have the autophagy. So in the autophagy, what happens is the lysosome can break down the organelles. Again, after breaking down, you will have a lot of things over here. So over here, you have the mitochondria, and once the lysosome comes and attaches to this particular vesicle, after that you can see. Okay, I have seen that somebody is raising their hand. I will take the questions at the end. Okay, so just please take your. Otherwise, I don't have any slides to discuss anymore. I mean, maybe about 10 minutes, I will be done. And then you can ask me questions. But you can keep your hand raised, and I will answer again. Answer your query again. So, yeah, I mean, in the autophagy, basically, you can see that there is a breaking down of the organelles. And again, when organ is broken down, cell might reuse few of these things, and the rest of the things can be excreted. So next, the part is we want to discuss is the cytoskeleton, and the cytoskeleton is basically, it can give the cell a particular structure. It can anchor few things, and it can hold the cell in its structure form. So what we see over here, this is the figure obtained, the image obtained with the fluorescence microscopy sensor, and there are different things which are tagged over here. In the cytoskeleton, there are mainly three things to study. One is hypotubule, other is hypofilament, and then intermediate filament. In this particular image, the microtubules are tagged green, and microfilaments are tagged orange, and DNA is in the blue, and intermediate filament is not tagged. So the main function of cytoskeleton network is basically to organize the structure and have different activities of cell, and this particular cytoskeleton can extend for the cytoplasm, and it can anchor few subcellular organelles, and it is mainly composed with the microtubules, microfilament, and intermediate filament. So now, particularly cytoskeleton can give mechanistic support to the cell, and it can, by giving the mechanistic support, the cell can maintain its shape. And especially it is very much important in the animal cell, because animal cell doesn't have the cell wall. If there is no cell wall, then it's very difficult to maintain the cell. So just hold on, okay, hold on. Yeah. So when you think about the cytoskeleton, it can provide the strength and resilience for and there will be few activities and few organelles that can anchor by cytoskeleton, and more, it could be more dynamic in animal, and it can quickly dismantle and reassemble in new location, and it can change the shape of the cell. So when you think about this cytoskeleton, we have already discussed that there are three main types of the cytoskeleton. First is microtubule, you can say it's a tubular structure, and inside it is hollow. In this particular figure, what you see is the fibroblast, and this basically has kind of cylindrical structure. It helps in maintaining the cell state, and it sometimes it will we can find this microtubules in the cell motility, especially in the cilia or flagella organelles, and it can help in the chromosomal movement, some organelle movement. The second type is microfilament, and over here you can see the microfilament has this particular shape, or you can say there is a intertwining things, just like hair bed, and with that it can support the cell. It can give the particular cell, and it is very when you think about muscle contraction and all those things, the microfilament will have something picture. Then it can help in the cytoplasmic spinning. Spinning is basically transporting few things from one point to other point. Then then you have the cell motility, and spinning is basically the microfilament. It can have very long length, and with that length, sometimes it's easier to move the cargo. I'll talk about that in a minute. And then it can microfilament, particularly it helps in the cell division. So apart from that, one more type is intermediate filament. So this intermediate filament is kind of in the fibrous nature, and if you think about the keratin protein, it will have this kind of shape. You can see there are fibers, and those fibers are kind of stuck together in chemical things, or you can say it will have the cable-like structure or spiral structure, and again it helps in maintaining the cell shape. Then apart from that, again it has very similar functions as other cytoskeleton types. I think one of the distinction is the intermediate filament can form the nuclear lamina. And lastly, I'm going to discuss about this intracellular cargo and how exactly, I mean, when you have something, protein, some protein layer, some other things synthesized inside the cell, you need to basically transport it to one point to other point. Um, in order to do that, we have specific motor protein, and this motor protein, that has receptor molecule or receptor motor protein. This is microtubule over here. What you see is microtubule, and these are the motor proteins which has kind of legs, and the motor protein is carrying the vesicle. So what happens is the motor protein uses the energy from the ATP, and ATP is hydrolyzed into ADP plus PI, and with that energy, the motor protein can walk along this microtubule, and by walking from one point to other point, it can carry this proteins inside the vesicle, and this proteins can be reached from one point to the other point. Um, it's a very unique situation over here, and when you think about the microtubule over here, you can see there is a image obtained with the scanning electron microscope, and this is this how the microtubule look like. These are the vesicles, and inside the vesicles there will be many proteins which can be carried from one point to the other. And with that, I think I want to end the lecture. If you have any questions, yeah, I mean, I'm ready to take the questions now. So, Samiksa.
Good evening, sir.
Yeah.
Sir, when we were discussing lysosomes, we said that it can do two types of functions, like autophagocytosis. Yes, sir. Yes, autophagy and phagocytosis. So, isn't there a third type in which the lysosomes engulf themselves?
Yeah, so this process over here, this is kind of engulfing over here. This is engulfing.
Sir, but here the lysosome isn't engulfing itself, like, so here it is engulfing different entities, right?
Yes. So what do you mean by this? I mean, can you elaborate a little bit?
Sir, we all know that lysosomes are known as suicidal bags because they engulf themselves sometimes.
Themselves? Engulf themselves?
Yes, sir.
That can happen.
So, is it a different kind, or it is like happens in these two?
So engulfing itself,
Yes, sir.
It is engulfing itself. It is what is basically it is digesting itself.
Yes.
So there would be one lysosome, and you can think that there would be bigger lysosome, and bigger lysosome is basically digesting the small lysosome. So it's the same process.
Yes, sir.
Okay.
Yes, sir. Answer one more thing. Could you please go to that endoplasmic reticulum slide?
This one?
Yeah, yes, sir, this one. So here it's written ER lumen. Could you explain the function of this part?
ER lumen.
Yes, sir.
Lumen.
Yes, sir.
So the lumen, here basically it's an enclosed space. So generic term. So over here you can see there are stackings over here, right?
Yes, sir.
And inside that stacking is basically the enclosed space, and that is called as lumen.
Okay, yes, sir. Thank you, sir.
Okay. Okay, next one.
Yeah, good evening, sir. Can you repeat that difference which you were telling, like in the case of cisternae in Golgi apparatus, the cisternae are stacked, while you were saying that like in one case they are connected and in one case they are stacked. So can you repeat that difference?
So one space is connected, other space is not connected. Can you repeat that one more time?
Sir, you were explaining something related to the stacking or the connectedness of the cisternae in case of Golgi apparatus. So can you repeat that point?
Yeah, sure. So now, Okay. So now you think about this Golgi apparatus, and let me go back to this, this figure over here. See, you have, at the center, you can say, but it's not exactly center, towards the center, you can see there is a nucleus. And, just to adjacent to the nuclear envelope also, you will find this endoplasmic reticulum. Now, endoplasmic reticulum is basically, you can say a lot of biosynthesis is going to happen over here. And after biosynthesis, let's say protein molecule, what is the molecule is there? It has to be transported to its particular place. How does it going to get transported? So what happens is, next to this endoplasmic reticulum, you have the Golgi apparatus. So, this Golgi apparatus, what happens is, again, there are two types. Basically, when the cistern from over here, this transitional cistern over here, transport vesicle, which is coming from the endoplasmic reticulum, that will attach to this Golgi apparatus, and it can be moved along this, the outer surface. Okay. So basically, this, Golgi apparatus basically serves some kind of, you can say it's some kind of mechanism from where, the particular proteins can be come together, and then it can be transported, and, then what happens is, at the end, the cistern will form with the cistern. Again, the vesicles could be formed, and these vesicles will be transported with the mode of protein to its destination. Is that clear now?
What do you think about this? Like, in one case they are stacked, and then in another case they are connected. And I do not understand these two terms.
Okay. I got your point. So now you look over here. This is Golgi apparatus, right? Now you see these are, this system over here. These are stacks. These are different entities. But in case of endoplasmic reticulum, it is just zigzag. Everything is connected over here. It appears to be stacked, but then, somehow with the zigzag pattern, you can see that it is connected. The connectivity is not direct, but there could be indirect connectivity. But in case of Golgi apparatus, these systems are kind of, different. These are just, stacks. Can say it's kind of, cylindrical object, and it has its own pocket, and within that pocket, the molecule can fit, and then after maturation, it will have, some kind of vesicle formation, and these vesicles will be, the inside vesicles, there will be some proteins or some other things which have to be transported across the cell.
Okay, sir. Okay. And sir, I had another doubt. In the slide about the cytoskeleton, there was a point which said that cytoskeletal elements oblique structure in prokaryotes, and there it was written homolog. So what does this point mean?
Okay. This cytoskeletal element oblique structure in prokaryotes. Okay. Again, over here, what we want to consider is about, so here, the homologs would be something like this. Cilia.
Okay.
I mean, overall, the cytoskeleton structure will be very much similar. But when you look into the minute level, in the minute level, if you want to study that, what might happen is the amino acid sequence of those, this particular when you think about the polymer, this polymer content, the polymer content would be very much similar, but not exact sequence. The sequence may not be similar. Or sequence may not be not similar. Sequence may not be identical, but sequence may be similar, and that is something called as homolog.
Okay, sir. And sir, another doubt, Yeah, it was written in that electron microscope, like there were images using light microscope and electron microscope. And it was written that electron microscope is used for dead cells and this light microscope is used for living cells. So why is electron microscope used for dead cells? Like, will it harm the living cells?
Okay, that's a nice question. See, it's a sample preparation method itself. Um, when you think about light, the light cannot basically, we are basically sitting and you can see, there is a lot of light around, and this light is not affecting us. Okay, it is not harming you, and so since it is not harming, which means that one can study the live cells inside the with the light microscope. But in case of electron microscopy, the sample preparation technique is altogether different. The electrons are very high energetic. Just like you think about the radioactive sample, or you can say X-ray. So we cannot go for repetitive X-rays. We cannot go for repetitive NMR. We cannot go for repetitive CT scan, because it will harm the cell. Altogether over here, when you prepare the sample for transmission electron microscope, what you do is, by default, the cell will be dead in the sample preparation itself. Without that, you cannot put it inside the microscope. So it is a way together different situation. There could be some other alternatives, but then I don't want to discuss about it over here.
Okay, sir. And since as you told that, like in prokaryotes, they have a small cell size and also the volume is small and the surface area, so the ratio is fine. But in case of eukaryotes, now as the volume increases, so in order to increase the surface area, to make that ratio, to maximize that ratio, then the organelle, they have a surface, they have a coil surface, or they try to maximize the surface area. But like as you told us, the surface area is the one through which the cell is interacting with the environment, right? Because it is exchanging matter and heat. So even if there is more surface area for the organelle, but the cell surface area is still the same, because the cell is overall enclosed and the organelle are inside the cell. So how does an increase in the organelle surface area affect the overall surface area by volume ratio of the cell? Because the cell surface area is the outer surface area, right?
I got your question. So now let's just try to compare with the prokaryotic. Okay. So prokaryotic, as such, there are no compartments, right? Everything is open. There is no compartment. Everything is open. So they can interact. All these cells. All these things can interact with each other. But in this case, in the prokaryotic eukaryotic cell, the cell size is much bigger. And with that, it has its compartmentalization. And now there are two comparisons over here to things. First comparison is the size, overall size. The second comparison is inside the eukaryotic cell. So now roughly, I want to maintain the same metabolic rate of prokaryotic cell versus eukaryotic cell. Metabolic rate is over here, there are a lot of interactions. Just like over here, the endoplasmic reticulum, it is, producing so many, the ribosomes are sitting over there, there could be other synthesis happening. So this is all metabolic activity. Okay. Then there are lysozymes basically, they are digesting food, and many things are there. And then, mitochondria is there. We have not talked about it, but then we might talk about it later at the later stage. So now, inside the cell, you want to have, let's say, very much similar metabolic rate as prokaryotic. Then how do you do that? For that, the eukaryotic cell has this strategy where each organelle has some kind of curvy nature or tubular nature, so that it can expose to a larger surface. And with that, many particles or many interactions can be happen. If that wouldn't have been there, then it would have been pretty much mess. Okay.
Okay, so sir, in prokaryotes, since the organelles are not enclosed, so they are free to interact, but in case of eukaryotes, since the organelles are enclosed, so in order to reach that level of interaction, they have this structure, they try to maximize the surface area. Right.